Can you all see classification of organisms? Somebody say something.
Yeah.
You can see it.
Yes, sir.
All right, then we'll start. Many different types of organisms out there, over 1.8 million species have been identified. You can check into a catalog of life if you want to view them. But there are probably many more species unidentified. A lot of them are in the soil, and I'll say a few words later about how you find out what's in the soil that is otherwise unidentifiable. So how do you keep track? There's so many different organisms out there. How do you keep track of them all? Well, we classify. We divide them into groups that show relatedness. And then within each group, each individual species gets a unique, unambiguous name. So the question is, how is that done? Well, people have been trying to classify organisms for a long time, going back at least as far as Aristotle, who lived from 384 to 322 BC. He looked at the world around him and categorized things based on their observable characteristics, the things he could see. So it was obvious to him that there were animals and there were plants. And the plants were easy. He classified them on the basis of size: big ones, medium size, and small, trees, shrubs, and herbs. Animals were a bit more complicated for him. So the first criterion was whether they had blood, at least what he perceived as blood. So those that had blood gave live birth. Those were mammals. Then there were birds, and there were non-flying egg layers, which were the amphibians and the reptiles. And then he had whales and fish. Now, those that had no blood, in his opinion, were things like octopus and squid, crustaceans like crabs and lobsters. He distinguished between those groups. Insects, flies and mosquitoes. He also classified scorpions and spiders as insects, although we know now that they're not. And then there were the shelled animals like snails and starfish. And then he had a category for what he called zoophytes, animals that looked like plants, so corals and anemones. So that's the way Aristotle did it. And this type of classification went on for hundreds, actually thousands of years. And it wasn't until Linnaeus came along that we really got a systematic look at classification. He proposed a binomial method of naming organisms. Binomial means two names, and the two names are the genus and the species. And this is the formal scientific name. And as you'll see in a minute, he established a formal hierarchical system to classify organisms. So what is the binomial for? Humans. This is Catherine Netti's daughter. Homo sapiens. This is Christina. Catherine Netti is the chair of the biology department here. What's the binomial for your cat? Felis catus. It's a binomial for a killer whale. Orcinus orca. So when using a name, it is written in the form genus, capitalized, and species, lowercase. So for example, the bacterium, Escherichia coli, you capitalize the first letter of Escherichia, and the first letter of coli is lowercase. And the genus and species names are either always either italicized, or if you can't italicize, for example, if you're writing out something in longhand, you underline as separate words. So if you do underline, you don't underline the space between the genus and the species. So here, Escherichia coli, italicized or underlined. And once used in a paper or any kind of a document, the scientific name, the binomial, is abbreviated after its first use. So for example, you abbreviate Escherichia as just E and then coli. So here's an example. Streptococcus pyogenes is a bacterium that is known to cause strep throat. The antibiotic amoxicillin can be used to kill most strains of S. pyogenes. So the first time you say the name, you spell it out, both the genus and the species. All subsequent times it's mentioned, you use just the first letter of the genus, S. pyogenes in this case. Is that clear? Everybody understand how you use a binomial, how you write it? Make sure you do, because it will probably come up in the future. In fact, I'm sure it will. So if you do have questions about it, please ask. Now, humans, we naturally classify things. Just think about everyday life. Think about what are the things that you classify every day?
Professor, you may have somebody waiting in your waiting room.
Okay, let me look. Yeah, thank you. Okay, you all have a place to live. You look around. How do you classify the things in your apartment or your house?
Hello, teacher.
Say your name.
Mastura Kayumi.
Yumi, okay. Mastura, how do you classify things? I'm looking behind you.
Yeah, because I was sick, teacher. Still, I'm not okay, but I want to take part in the class.
Okay.
I got severe pneumonia after that COVID-19.
You got coronavirus?
Yeah, because
Oh,
my husband got
I'm
it.
sorry to hear that. I hope it's not affecting you badly.
Yeah.
But I'm looking behind you, there's a bookcase, so I can classify something in your house as books.
Yeah, I have some books.
And I can classify other things in your house as furniture. So anybody else, how do you classify something that you encounter all the time?
This is Zach Nuschler.
Uh-huh.
And electronic devices.
Good. Yeah, we all have computers, so what else? Go outside. Haley, Garrett, okay.
Yeah, I was thinking like of food, how we classify it between like healthy and not healthy.
Yeah, we classify food in a lot of different ways. Fruits, vegetables.
What about cabinets?
What about what?
What about cabinets?
John Riley, just a minute, I gotta write your name down.
This is Marissa. Would you classify something as hazardous or non-hazardous?
Sure. Yeah, you guys are doing good. Just keep thinking. What it amounts to is that we classify things all the time as we go through the course of a day. Anybody else can think of something that you classify?
Classify things you like and don't like.
That's good. Don Edwards, was that? Edwards, okay.
Terrence Belprez, school and work. Sometimes it ends up being both.
Okay, good.
Dylan Strangman here. I was thinking about movie genres.
Sure, yeah, that's a good one.
Bowie
Ariana.
John here, and you can
Okay.
classify things that are painful and comfortable.
Painful versus comfortable, yeah.
Marissa, again, you can classify something as dead or alive.
Yeah, that's true. And we do that all the time. You see a bug, it's either a dead bug or a live bug.
And we do that a lot with the plants in our front yard.
We need rain, Scott.
Apparently plants like rain.
Yeah. Their vacuoles run out of water after a while.
I have a hypothesis about that.
Uh-huh.
Yeah.
Okay,
There's a
well,
theory.
Teacher, I have also an idea about the classifying the information. When we experience something that are bad things or that are good things, we can classify it by our experience.
you mean like
All
enjoyable?
the things that we have some experiments about something, we can classify the information.
Yeah, yeah, that's a good example. Okay, so the point here is that we're constantly classifying things because we like to relate one type of thing to another. So you distinguish between items of furniture and books, for example. And we do that all the time unconsciously. So many organisms, if we went back to the way Aristotle and many other people in ancient times, they classified things pretty much like we're doing right now. If they look alike or have similar function, then we put them in the same category. But the way we classify living things now is based on their molecular construction, in particular, ribosomal RNA, and I'll say a little bit more about that later after we talk about the gross structure of living things. So the current system of classification has three broad categories. These are called domains. These three domains are shown here: bacteria, archaea, and eukaryotes. The eukaryotes are divided among plants, animals, fungi, and protista. The protists are sort of a grab bag of anything that doesn't fit in the other three categories gets classified as a protist. Now they're usually small, usually unicellular. So how do we distinguish, or what's the difference between bacteria and archaea? Well, they're similar in a lot of ways. Even though they're different domains, they're all small, they're prokaryotes, they're all single-celled. They all reproduce the same way by a process called binary fission. They all have ribosomes and a cell wall, and their genome is just DNA. It's called a nucleoid. So how do you tell archaea and bacteria apart? Well, they have different structures to their cell walls. Archaea have a pseudopeptidoglycan wall, whereas bacteria usually have a big component of lipopolysaccharide. Archaea, probably the most distinguishing feature of archaea is that they live in harsh environments. like hot springs and salt lakes, whereas bacteria never occupy those kinds of habitats. They live in temperate zones, temperate conditions. And another distinguishing feature is the archaea never produce spores. They just go on dividing. They don't have a dormant phase, whereas bacteria, many bacteria, produce spores which are dormant and allow them to live for long periods of time while being inactive. So I just wanted to make the point that these two domains, even though they're called different domains, they're very similar in a lot of respects. So the way we classify is to first put things into big boxes and then divide those big boxes into smaller boxes and keep on going. So if we take the domain eukaryotes, there are 4 kingdoms: the protists, the fungi, plants, and animals. So we've divided one box into 4 boxes. And then within, if we take just one of those kingdoms, the animals, there are what are called phyla. There are 8 different phyla, which distinguish various different types of animals. And so if you go through the whole identification, you start with the domain, then that gets broken down into kingdoms, phylums, we just did, then classes, orders, families, genera, the plural of genus is genera, and species. So let's go through a classification. We have 3 different species here: species 1, species 2, and species 3. If we look at the domain of these 3, they're all eukaryotes. For kingdom, they're all animals. They all fall in the animal kingdom. Phylum, they're all arthropods. When we get down to the level of class, we start to see differences among them. Species 1 is a crustacean, and the other 2 are insects. And we're down another level to order. One is Decapita, and the other 2 are Diptera. And then when we get to family, we find that these 3 species are in 3 different families: Caridae, Drosophilidae, and Nematocera. And then we come to the real identifiers, the binomial, the genus, and the species. So species 1 is Homerus americanus, species 2 is Drosophila melanogaster, and species 3 is Aedes aegypti. Now, what are these? Homus americanus is a crab. Drosophila melanogaster is a fruit fly. And Aedes aegypti is a mosquito. Okay, now you have to brace yourselves for my joke slide.
Oh boy.
Anybody get it? Scott doesn't like it. Well, the reason I bring this up is because fruit flies came up here, and fruit flies are the favorite organism of many, many geneticists. And as you go around, especially in university labs, and you go into a Drosophila lab, and you almost always will see this sign: time flies like an arrow, fruit flies like a banana. What does it mean? It's nonsense, and that appeals to geneticists. And then there's always usually some wise guy that will pervert something like that. As you see here on the right, time flies like an arrow, fruit flies like a banana, house flies don't like going flat. That means they don't like to be swatted. All right, that was my attempt to amuse you. And now we'll go on. Okay, getting back to criteria. Currently, classification is based on phylogeny. And phylogeny indicates the evolutionary relationships between organisms. In other words, how far back do any two organisms have a common ancestor? And phylogenies are based on the genetic code of ribosomal RNA. And we haven't talked about nucleic acids yet in this course, but we will. And for right now, all I need to say is that the sequences of bases in RNA, particularly ribosomal RNA, they're highly conserved throughout evolution, and they act like a biological clock. So we have pictures here of 3 organisms that fly. So if you were just observing things, you might say that these are all highly related because they all fly, they all have wings. But if you look further, even just structurally, you can see that a bat and a bird, they both fly, and they're evolutionarily homologous. That is, their wings are both derived from their legs. Whereas an insect has no evolutionary commonality with birds and bats. Their wings are made from completely different parts of their bodies. So if you look at this little diagram here of the evolution of organisms, at some point in evolution, 4 limbs evolved. And the 4 limbs evolved into wings, in the case of bats and birds, did not evolve into wings in the case of mice and crocodiles. Yet, phylogenetically, in other words, in terms of evolution, bats and mice are much more closely related to each other than bats and birds are. Birds are more related to crocodiles. So what this is getting at is the difference between analogous structures and homologous structures. So analogous structures have a similar function, but no evolutionary similarity. So we're talking about wings, a similar function. Bird wings and insect wings have similar function. But homologous means similar evolutionary history. In other words, that they have a common ancestor somewhere back in their heredity. So bird wings and bat wings share an evolutionary ancestor. You can see here in this top diagram, the legs are modified in both in order that they can fly. So homologous structures, this is a reiteration, bat wings and bird wings are homologous structures. They share a common evolutionary past. Analogous structures, the wing of a honeybee is similar in shape to a bird and a bat wing, and it serves the same function, but it's not composed of bones and has a distinctly different structure and embryonic origin. So these types of wings, insect versus bird and bat, illustrate an analogy. Similar structures do not share an evolutionary history. So is that clear to everyone? Understand the difference between analogous and homologous origins.
Yep.
Everybody got it?
Yep.
Okay.
Clear as mud. Let's move on. That was a joke.
Yeah, well, you got as many laughs as I did.
Darn right, man.
My joke. Okay, I guess we're going to have to learn how to tell jokes in order to get this class to laugh. Okay, contemporary bases for classification of organisms. Morphology plays a small role, but ribosomal RNA sequences have molecular precision. So why is ribosomal RNA so useful for classifications? Well, number one, it's ubiquitous. It's found in all organisms, both prokaryotes, eukaryotes, bacteria, animals, plants, whales, trees. Everybody has ribosomal RNA. Within the sequence of ribosomal RNA, some regions are highly conserved and some are highly variable. So if you look at this structure on the lower left, it's the sequence of a particular ribosomal RNA. The red residues are highly conserved, and the blue and purple ones are highly variable. And so you can use highly variable ones to discriminate among species, and the highly conserved ones are reference points for analysis. And if you look to the right, you can see that the ribosomal RNA sequences of human, mouse, and rat are compared and shown relative to bacteria, E. coli, S. marcescens, and B. subtilis. And you can see that the yellow ones are conserved residues throughout. They're probably representative of living things in general. Whereas if you look at green or red, they are showing differences between mammals and bacteria. So just by sequencing, that's how phylogenetic trees are constructed, like this. So there's a common ancestor down here in the black, the original founder cell that probably evolved in the ocean somewhere, gave rise to all life forms that we know now: the bacteria, the archaea, and the eukarya. Now, the bacteria, most of them are non-photosynthetic, and they're single-celled, as I said. Some of them do have chlorophyll, like the cyanobacteria and the green filamentous bacteria. So they do produce food, but most of them consume food only. And what distinguishes them from the archaea is that I went through that earlier. You can refer back to it. But in general, the archaea exist in extreme environments. So for example, the halophiles grow in highly salt, like salt lakes. The methanogens either can grow on methane or they produce methane. The thermoproteus type of archaea live in hot conditions. I once worked in a biotech company that collected environmental samples, and we had a bacterium that was collected from a hot spring that could only grow in boiling water. If you reduce the temperature below boiling, it stopped growing. And then we have the eukarya. The ones we're most familiar with are the animals and plants, and perhaps some fungi. And all the rest of these are things that we rarely see. So consider the domain bacteria, the simplest organisms. They're prokaryotes. The three main shapes we talked about earlier, the spirochetes, the rod-shaped bacilli, and the cocci, the spherical ones. They're found everywhere on the planet, even in the polar ice caps. Most are environmental organisms, and many of them are human pathogens. For example, one of the spirochetes causes syphilis. Domain archaea. I've compared them to bacteria in an earlier slide, so this is mostly redundant information. They're single-celled. They are ubiquitous on the Earth. They're not, most of them cannot be cultured. In other words, you take them into the lab, try to grow them in a petri dish, they won't grow. So their existence is known from metagenomics. Does anybody know what metagenomics is?
with different genomics.
Metagenomics.
Meta another means.
That's what I'm asking. Just curious if anybody knows. There's no particular reason why you should. Metagenomics is an analysis of genomes in the environment. So, for example, you might scoop up a handful of soil and extract the nucleic acids from that, and then sequence all those nucleic acids, and then start comparing the sequences. And the sequences will join together and reveal the existence of organisms. So thousands of new organisms have been discovered by looking at environmental samples like soil, like a termite gut, any raw sample from nature can be extracted and the nucleic acids sequenced. So that's how we know about many microorganisms that won't grow in a lab. None of the archaea are known to be pathogens of anything, although they are hosts to viruses. And here you have pictures of thermophiles grow in hot places. Acidophiles grow in acidic environments, and halophiles grow in salty environments. Shapes of archaeal cells. There are various different shapes. Some of them are pretty odd looking. And then we will go to domain eukarya. Contains organisms composed of eukaryotic cells, animal cells, and plant cells from 10 to 100 micrometers. And we've already talked about the protists and the fungi and the plants and the animals. We have pictures here. Which of those pictures is animal?
Esteban Monzon.
The first one.
Yeah, the first one.
Top one.
Yeah, the first.
Okay, that was Esteban. And how do you know?
The bottom one has more of like a square shape. So, and the one above is kind of just like they're all clumped together. There's no real pattern or shape to them.
Yeah. And what does that, excuse me? Go ahead.
The second one like a boxes. It's the shape, the cell shape like boxes.
Yeah. And so structurally, what's the difference that accounts for this difference in shape?
Esteban speaking again. The first one, the reason why the second one has the shape is because of the cell wall.
Yeah.
So, yeah.
The first one does not have a cell wall.
Yeah.
Okay, good. The protists, as I said earlier, if it doesn't fit into any other category, it's lumped in as protist. So examples would be amoebae and volvox, which you find in the ocean. More examples like kelp, algae, oomycetes. You may, in labs that you've had, may have looked at paramecia and amoeba, and trypanosomes, which are parasites. Okay, fungi, a very diverse group. Fungi are interesting organisms in that they don't have internal stomachs. Their stomachs are actually outside their bodies. And by saying that, I mean that the only way they can get food is by secreting enzymes that break down food that's outside their cells. And then they absorb the food that they've digested digest it into small molecules, take it up and use it for food. So that is the function that they have. Instead of a stomach, we have the enzymes that break down our food located in our stomachs and digestive tract. This particular fungus is Penicillium. Somebody mentioned that earlier in class. It's the fungus that produces the antibiotic penicillin. These little blue balls you see are the spores, the asexual spores of this fungus.
Good question.
Yeah.
Where does the enzyme secretion happen? Does it happen in the mycelia, or does it happen also from the fruiting bodies?
It's largely from the mycelium. Most of the body of a fungus that you can see, or if you look at a microscope, is the mycelium. So the mycelial cells produce enzymes, secrete them, digest whatever they're next to, and then take up the digestion products. So the spores, which you see here, are dormant until they're put in a warm environment. They just sit there. Once they have water and a warm place, they'll germinate to make a mycelium, and then the mycelium can absorb more nutrients.
Is that when penicillin is created? How is that created?
Penicillin is made by the mycelia, and it's secreted just like the enzymes are. Many small molecules are secreted. Penicillin is a small molecule. It's a secondary metabolite, and that's how Alexander Fleming first noticed the effect of penicillin. A fungus was growing, the mycelium of the fungus was growing on the agar, secreted the penicillin, which diffused out into the agar and killed the bacteria.
Okay, thank you. I get it.
I should get a picture of that to illustrate the point. It's really quite dramatic. You all see fungi. On the right here, has anybody seen anything like this in their refrigerator?
I try not to.
Want some bread?
Yeah.
Would mold be classified as a fungi?
This is a fungus. It's a very common fungus. It's ubiquitous, found everywhere. It's an aspergillus species, and it's all over. So I don't know, these are probably oranges. So if you go to the store and buy oranges, it's got aspergillus all over it and spores, and probably penicillium spores too, as well as a number of other species. And as long as they have nothing to grow on in the peel of the orange, protects the orange from attack. But in this case, the orange started to rot and secreted its insides, and the fungus immediately, the spores germinated, and it grows all over and then accelerates the rate of rot.
So I have a question. So if you have bread and it has mold, like a spot of mold on it, if you rip it off, is it still edible?
In general, yes, but I don't recommend doing it because there is a chance that the fungus has produced toxin that's used into the fruit or the vegetable, and you don't want to eat that.
And bread's relatively cheap, so.
Facts.
We don't have time in this course, but fungi are famous for producing what's called mycotoxins, which are fungal metabolites that contaminate food. Okay, we have another interesting fungus here on the left. It's called a nematode trapping fungus because it makes traps. You'll see here in the center of the picture, there are 3 cells in the middle of a hypha, and those 3 cells form a circle, which is a trap. This fungus lives in the soil, and that's where nematodes, little worms, also live there. So if a nematode just happens to run into this trap, as this one has, the trap snaps shut, holding the nematode in place, then these trap cells start to secrete enzymes, and they digest the nematode, and the digestion products of the nematode are taken up by the fungus. It makes more mycelium, so it can make more traps and catch more nematodes.
So a plant analog of that would be like the Venus flytrap or the pitcher plant.
Yeah, yeah, same idea.
No.
All right, fungi help us out a lot. They are involved in bread production. What kind of fungus is involved with bread? Well, it says so here, yeast. Yeast are fungi, and they're very useful for making beer, wine, and bread. Cheeses are made, many of them are made with, not with yeast, but with filamentous fungi. So the same genus that makes penicillin also helps them to make cheese. Penicillium camemberti makes camembert cheese. P. roquefortii makes Roquefort cheese, and that you can see here on the right. Then some fungi we actually eat, like the ones you see at the bottom, and these are the fruiting bodies. These are the sexual structures of the fungus, not the main body of the fungus. The main body of the fungus is just little threads that will occasionally undergo sex, and we'll talk about that later in the course. Okay. So any more questions about how fungi? Notice the binomials here for penicillium. I spelled it out in the first case, and in the second case, since we already had penicillium, you know the P stands for penicillium. So plants, we're going to spend a lot of time later in the course talking about photosynthesis. That's the main interest that we will have in this course in plants, because plants are the source of all food, green plants. If it weren't for them, life wouldn't exist as we know it on this planet. It would all have to capture energy in a different way, not from the sunlight. And finally, animals. We went through all the 8 phyla of animals earlier. We will talk about animal cells later in the course when it comes to respiration, how do animals and plants process their food, derive energy from food. But that's all for later in the course. Won't talk more about that now. And I think, yes, that's the end of this slide set. So I'll take any questions or comments that you have about this topic or what I've said before we go on.
I have a quick question. Dylan speaking.
Okay.
I noticed for a couple of the binomial names, they'll just have the same species name as the genus. So like, for example, there's a tarantula that I know called avicularia. Why is that?
It has the same genus and species name?
Yeah.
That's the choice of whoever named or discovered that organism. For some reason, some people will do that. They'll give the same name to both the genus and the species. To me, that's sort of unsatisfying, but whoever chose to name it chose to do that.
Okay, thank you.
That's one perk that you have as a scientist who discovers things. If you discover a new organism, you can give it a genus and species name. If you discover a new gene, you can name that gene whatever you like. Anything else? Okay, I will stop this, and I have enough time. Since you have a 15-minute quiz today, I will take 15 minutes off the class time.
Is the quiz gonna include everything we're learning today or just?
No, the quiz covers just the topics that we did last week, the methods of scientific inquiry and the introduction to biology. If you study those topics, you will be prepared for the quiz.
Yeah, teacher, it's possible that I do the quiz later because I want to read it again, the slides from the PowerPoint that you.
Okay, you can do that. You have until midnight tonight, so you can
Okay.
review the
Thank you.
slides and take the quiz later today.
Okay, thank you, teacher.
Anything else?
Excuse me, Professor. Do we have writing in the quiz?
Have what?
Writing.
Essay, very short, just a sentence or two.
I think 15 minutes is too short.
Have you taken the quiz yet?
The first quiz, I took the first quiz. No, the second one, no.
Okay,
But for writing, I take more time.
There's not much writing involved. Tell me, after you've taken the quiz, let me know if it's not enough time.
Okay. Thank you.
Anything else? All right, I'm going to share.
We had another quiz also before this one.
It was a practice quiz.
Yeah.
If we've already finished the quiz, are we allowed to clock out early, or do you have something at the end of class?
So what's your question?
Like, we've already finished the quiz. Are we allowed to leave class, or do you still need to use that time? Like, you have an announcement when we're done taking the quiz.
No, I have another slide set here on life in the universe that I'll try to fit in the next 10 minutes, but if you need to leave before that, that's fine.
Okay, thank you. That does answer my question.
Okay, you should see life in the universe, do you?
Yes.
Yeah.
Okay. So this is not really a part of the curriculum for this course, but it's a topic that fascinates me, and I'm sure that probably most of you have wondered, is there life anywhere else besides the planet Earth? So these are just a few thoughts about that. Just to remind you, this is the scale of things. We go all the way from molecules to giant pine trees. And we talked about how we visualize all these things, but then to get beyond life on this planet, we use instruments like telescopes, including space telescopes. And that prompts us to ask, among those multiple billions of stars out there, we know that most of them have planets. So what is the probability of life on planets in other parts of the universe? What are the conditions that are required for life? Some people spend their entire careers addressing this question. And in general, people who do, they're called exobiologists, they assume that for life to exist, liquid water is necessary. And there has to be an atmosphere that contains, at the least, nitrogen and carbon dioxide. Now this is a picture of the Kepler Space Telescope. It was launched in 2014 and retired in 2018. The goal of this telescope was to get out away from the planet Earth, get away from the atmosphere, which interferes with taking pictures, and discover planets in other parts of the universe. And so here are some of the Kepler planets that were observed with the Kepler telescope. And it's plotted against planet temperature and size. Temperature and size are critical for whether life can exist. So if we assume that for life to exist, it has to have liquid water, you look down here in the blue region of this thermometer, water freezes at 32 Fahrenheit and boils at 212. So life has to exist somewhere. So the planet has to have temperatures somewhere in that range. And you can see that Earth does. Elsewhere in the universe, it's way too hot for life to exist. And because water can't exist as liquid, it boils away. So there's what's called the Goldilocks phenomenon, the Goldilocks conditions for life. You remember the story of Goldilocks? One was too hot, one was too cold, and one was just right. Well, that's true for planets. For a planet to sustain life, it can't be too hot or too big. It can't be too cold or too small. But rather, by those parameters, it has to be just right. So Kepler found a planet in a system, in a solar system known as Kepler 186. And this picture shows a comparison between Kepler 186 and our own solar system. The planet most likely to have life is called planet F, which is what, the 1, 2, 3, 4, the fifth planet out from the sun in the Kepler system. The sun is somewhat smaller than our sun, and so it gives off less heat, but F is at just the right distance for water to be liquid on this planet. If you compare that to Earth, Earth is the third planet out from our sun. So the conditions for life to exist are liquid water. An atmosphere is likely a requirement. Another requirement is probably a liquid core that generates a magnetic field. And the reason that's important is that deflects radiation coming in from elsewhere. And for us, that would be primarily our sun, the solar wind. But if it wasn't for the liquid core of our planet, life would all be destroyed by radiation. And then finally, the size of the planet, the mass of it, is critical because the mass determines the gravitational force. So if the planet is huge, the gravitational force is enormous and would crush living things. If it's too small, the mass is too small, it can't keep its atmosphere. The atmosphere escapes into space. So the size of the planet is also critical to satisfy the Goldilocks conditions. Okay, this shows the point of this slide is to emphasize distances. Up in the upper left, I say the main problem in finding life elsewhere is distance. So if we think in terms of the speed of light. The distance from the Earth to the moon is 1.5 light seconds. In other words, 1.5 seconds for light to get from the moon to us. From the sun, it takes 8 light minutes. In other words, eight minutes for light to travel that distance. From Earth to Mars is 12.5 light minutes. And from the nearest star to us is 4.3 light years. So that's a huge distance. And from Earth to the other side of our Milky Way galaxy is 52,000 light years. Now Kepler 186f is 500 light years away from Earth. So that's 2.5 times 10 to the 15th miles. Huge distance. Yet it is still in the Milky Way galaxy. And the time to get there, if we were going to leave from Earth and try to go to Kepler 186f, if we were traveling at the speed of light, it would still take 500 years. But we don't know how to travel at the speed of light. So if we used the best rocket that we're capable of building now, so that we could get away, have enough power to get away from our sun's gravity. In that kind of rocket, it would take 242,000 years to get to Kepler 186f. So just the distance alone probably precludes us from ever really analyzing, studying this planet, even though it seems to have the conditions for life. This slide is trying to make another point, and that is, if there is life, say on Kepler 186, what form would it be in? And I've gone through a bunch of calculations here about how life would form in a prebiotic ocean. It would take billions of years to finally get to some primitive form like trilobites, it forms in deep ocean vents. I'm running out of time, so I'll try to speed along here. So highly complex life forms have occurred only in the last few hundreds of thousands of years on Earth, 0.0001% of life's history. So the conclusion is that any form of life on other planets is very likely to be primitive, and that's illustrated here. If we assume that life on other planets happens, or it has happened like it has on Earth. So our Earth is 4.5 billion years old. The earliest life forms probably occurred about 3.5 billion years ago. And they went on living and living and living for billions of years until we started to get photosynthetic multicellular organisms about 2 billion years ago, more complex organisms 1 billion years ago. And by 600 million years ago, there were complex life forms in the ocean. At 360 million years ago, there were complex life forms on land. And only about 7 million years ago did primates, in other words, intelligent life, occur on this planet. So the point is that for billions of years, there was life here, but it was unintelligent. It was just cells or unintelligent other forms of life. So humans have been here for only 0.4. So 7 million years is this arrow clear at the end. Humans have been here for much less than that, 0.4 million years, which means it's practically on the zero. So the likelihood of life is much higher than that for intelligent life. So the scale shows that life went on for billions of years. If an alien were to have come to this planet looking for intelligent life, it wouldn't have found it. It's only been in the last very, very short time that life on this planet has been intelligent enough to be self-aware. So the conclusions that I draw, these are my opinions, they're not really conclusions. Is there life elsewhere beyond Earth? To me, it seems the probability is very high. Why is that? It's because the number of Goldilocks planets in the universe is probably astronomical. That is, the number of planets associated with stars that have conditions on which life could form, probably a lot. But the likelihood of detection by us, very low. And why is that? Because of distance and because of the form that life is probably in. The distances are too great, and life forms at any point in time are likely to be too primitive to respond to a query by people from Earth. So the question then is, will we ever detect extraterrestrial life before we ourselves become extinct? And we will, we don't know when. At the rate we're affecting climate, it's worrisome. But at most, we're not going to last forever. So if our species is going to continue to exist after our planet is uninhabitable, we're going to have to escape from our planet. Space stations, you can use your imagination to think about that. At any rate, those are my musings about life beyond the planet Earth. Any questions or comments about this? I'm sure some of you have opinions. Hearing nothing, we are out of time. So those of you who haven't taken the quiz yet, you have until midnight to do so. And I will see you again next Monday.
Thank you, Professor. Bye.
Have a good weekend.
Have a good day, Professor.
Thank you, Professor.
All right.
Thank you, sir.
Okay, bye.
Goodbye. Have a good day.
Okay, thanks.